Okay, question 7, and our command word is contrast. So contrast is like contrast and compare. So you've got two things, DNA and mRNA. So I think you want to think about your structure as being DNA has this structure and mRNA has this structure. Okay, so every time you need to make both statements, if you just give me a fact about DNA, you won't get the mark. Okay, so first thing is DNA is double-stranded. Okay, I'm going DS. Whereas RNA is single-stranded. Now, we have mentioned that RNA genomes can be double-stranded RNA, but the question here is very specific about mRNA as opposed to tRNA, which has those double-stranded regions in them, whether it's a single-stranded molecule that has the hairpin loops, or ribosomal RNA. Okay, so read that question carefully. We've then got a size point. Okay, so DNA is longer. I think someone said bigger, but that's a bit of an ambiguous term. Whereas mRNA will always be shorter. Okay, we're then going to look at the bases. So think about thymine in DNA versus uracil in RNA. And then I've also mentioned the sugar difference there as well. Okay, and then DNA uses base pairing, whereas mRNA doesn't. And then this final point we actually haven't worked on yet. We're going to look at it a little bit later in the syllabus. But DNA has introns, whereas mRNA has no introns. Now, introns are regions within a gene that are not expressed. So what happens is DNA gets made into pre-mRNA, which is then spliced into mRNA. And splicing is the process of removing these regions called introns that are not used to make protein. We will go through it, don't worry, but there are 5 marking points there that you could have got for your 3 marks. For your revision, if you do not have it in your notes, I really suggest that you make a table with all of these DNA points on one side, so features of DNA, and then features of RNA on the other side. And that way you've got something really quick and easy to compare. And in these questions, you can just pop down your answers very speedily. Okay, next question. ATP is a nucleotide derivative, so it's not the same as as a nucleotide. It's derived from that nucleotide, so we've got another contrast. So we wouldn't say ATP is like this, whereas a nucleotide found in DNA is like that. So lots of people made a really good point that ATP has 3 phosphates and DNA nucleotide has 1 phosphate. And a second marking point that ATP uses ribose as the sugar, whereas DNA has deoxyribose. Okay, so we did look at that as well in our lesson. It's a little bit different. Finally, ATP is a very specific molecule because there is such a thing as CTP, GTP, and TTP, but ATP is the one we mentioned because it's the one that's made in respiration and used to transfer energy around the cell. So the base is always adenine, whereas in DNA, the base could be one of 4. So it could be T, G, C, or A. So it could vary. Okay, your mark scheme will accept diagrams. So if you had drawn a diagram, a labeled diagram of ATP with its 3 phosphates and its ribose sugar versus a labeled diagram of a DNA nucleotide with its deoxyribose and single phosphate, then you would have got 2 marks for that as well. Okay, so do use diagrams if you find them helpful, if you kind of find it easier to remember for these contrasts, but your diagrams have to be absolutely correct and on point to get those 2 marks. Again, another one where it might be useful to make some tables in your notes.